one of the things that you often think about is how can I enrich the quality of the structure, the water holding capacity, the drainage? How can I add more nutrients and organic matter? And how can I support the below ground organisms in my soil? So we find that cover cropping, there's a lot of research out there about the benefits of cover cropping, especially cover crops being used in open field systems. We see that it increases soil organic matter. It helps return carbon to the soil. It can also help with capturing some of that nitrogen that would have been lost. And then it can also help with reducing the need of fertilizers. So we wanted to look at how these cover crops would perform within a high tunnel system in both winter and summer seasons. So in our summer experiment, we had 8 treatments. We had buckwheat, millet, sorghum Sudan grass, a weed-free bear control plot, cowpea, and buckwheat, millet and cowpea, and sorghum Sudan grass and cowpea. So our cover crops were planted in, for the summer season, they were planted in June. And in the first year, they were terminated or cut down in September. And in the second year, they were cut down in August. And there was a reason why we changed it a little bit for the second year, and I'll share that with you in a few moments. So now for our winter experiment, we also had 8 treatments. We had rye, hard red winter wheat, triticale, weed-free bear control plot, hairy vetch, rye and hairy vetch, hard red winter wheat and hairy vetch, and triticale and hairy vetch. So for both the 2017 and 2018, we planted them in October, and we seeded them in October, and then we terminated them in May. So I'm going to show you a little bit of the results that we had from the winter and summer cover cropping to see the biomass that we accumulated within the high tunnel. So this is an overview of the summer cover crop biomass. As you can see in the left-hand column are the treatments that we had, and we have it divided by 2018 and 2019 because there were significant differences between the 2 years. So as you see here, the first year, the highest biomass accumulator was the sorghum Sudan grass treatment, and it was the same for the second year where it was the highest biomass accumulator. Now we changed the amount of time in which the cover crops were growing in the high tunnel between 2018 and 2019 because we were getting so much biomass in 2018. It became almost a horror. We would walk in and the sorghum Sudan grass it grows so tall that it was putting pressure onto the walls of the high tunnel. And we really didn't want to have to replace plastic in between our study. So what we did is we decided, how about we just reduce it by a month for the second year, and we can also see what that effect would be. So when we looked at that, we found that when it came to the biomass, yes, the biomass was significantly different between 2018 and 2019. However, the available nitrogen that was added to the soil, there was no significant difference. And that could be due to, in 2018, us having those lignified stalks, because as the sorghum was growing, it was actually becoming harder and more lignified, that it took longer for that nitrogen to become available once we terminated. And so the young tissue and the 2019 cover crops, they also had a lot of nitrogen available, that it was comparable to 2018. There was no significant difference within the soil total available nitrogen. Now, when it comes to the winter cover crop biomass, our winter cover crops did not perform as well as our summer cover crops. We had some freezing that actually happened where a lot of the cover crops really didn't get to take good root before the frost came. So a lot of them did not really recover by time of termination. So as you can see here, most of our treatments actually did produce biomass. However, like our hairy vetch treatment did not fare well in 2018 or 2019. We're not sure as to why hairy vetch did not perform well, but we've just realized that hairy vetch may not be the best candidate for cover crops, at least in Kansas and high tunnels. Okay, so now when it comes to below ground arthropod study, we had two research objectives. As I shared, just with that research that I showed, that was just some of the research that we did from that I brought in from the greater study. This below ground arthropod study was a complementary study to that study. And we wanted to look at the overall soil ecology and what was happening within the soils as we were terminating the cover crop and the weeks thereafter. So our research objectives were to monitor how changes in soil moisture affect arthropod abundance in high tunnel systems. We also wanted to determine how soil carbon, nitrogen, and organic matter reserves during winter and summer growing seasons influence arthropod abundance and family diversity. So our methods for soil sampling, I also need to share with you a little bit of the methods that we had for the greater study. So in the greater study, we also utilized similar methods. So we used a probe to take soil samples out of the high tunnel. When we were doing soil samples for total nitrogen, carbon, and organic matter, we went 6 inches deep within the soil. Whereas when we were sampling for gravimetric soil water content and for arthropods, we collected soil within the top 3-inch layer. And the reason why we collected soil from only the top 3-inch layer when we were doing the arthropod sampling was because that's where arthropods predominate, which is within the top 3-inch layer of your soils. As you go deeper and deeper into soil layers, you have fewer of arthropods. You'll still have other microorganisms such as fungus and bacteria, but arthropods usually reside within the top 3-inch layers. So when we took the samples at termination, 2 weeks after termination, 4 weeks after termination, and then subsequently 8 weeks after termination. So we had 4 time points in which the soil was sampled from each plot. All right, so when it came to actually isolating these arthropods from the soil, we had 2 methods that we used, and it was stepwise. We started off with using a sieve where we were able to put the bulk of soil within the sieve and was able to remove all those large clumpy pieces of soil. And that sieve is kind of like a colander or anything that you've seen where you can sift things out. And then the second step was for us to use a modified Berlese funnel system. What we did is we had these funnels, as you can see here, that were lined with tulle. And then the soil was put within the tulle lining. And at the bottom of the funnel were vials filled with isopropyl alcohol. And we would put a heat source above the funnels to help drive those arthropods from out of the soil into the alcohol. And once they were in the alcohol, we had them pretty much we were able to look at them in a microscope. They were no longer alive, unfortunately, but we were able to look at them on a microscope and we were able to classify them to families and classes. So our statistical analysis, we were able to get some help from Trevor Heffley, a statistician at K-State, but the statistical analysis that we used generalized additive models, and we had a couple of continuous predictor variables, which was soil total nitrogen, total carbon, organic matter, and soil moisture. And then we had a categorical predictor variable, which was cover crop type or treatment. I must share with you that we continued to keep it a 2-season study. We separated them by season. However, we did not separate them by year. We did not separate them by year because we didn't find any statistical significance between the 2 years. So we were able to analyze the 2 years together. And our significance level was a p-value that was less than 0.05. All right, so let's look at our soil total nitrogen. So when it came to nitrogen, carbon, organic matter, we sampled from the 6-inch depth within the soil. So as we see here, our winter soil total nitrogen, we can see that our total nitrogen peaked at 28 days post cover crop termination in year 2. And the bare control plot had the lowest total nitrogen percentage. There was a significant difference between the treatments, but we did find that we had this odd thing that took place where our soil total nitrogen, it looked as if it decreased within 2018, going from the day of termination to 8 weeks post-termination. We do not understand why that took place. That's something we still want to research and find out whether maybe the cash crops that were planted may have had any effect on this, but we haven't been able to research that and find out the answer for that. Now, when it came to soil total carbon, the total carbon fluctuated throughout cover crop decomposition. However, it was the highest at day 28 in both years, with rye and vetch treatment having the highest being the highest biomass accumulator. And it also had the highest amount of total carbon, the rye and vetch treatment. Now, when it came to the winter soil organic matter, unfortunately in 2018, we weren't able to get the last couple of time points. However, what we did see is that our cover cropping resulted in increases in soil organic matter in year 2 that were statistically different between the cover crop treatments. Similar to biomass production, the changes in soil organic matter was variable between the 2 years. It is still unclear why winter organic matter decreased in year 2 compared to year 1. So when it comes to soil total nitrogen, for the summer cover crops, we see that there was an overall increase from year 1 to year 2. And we saw that in year 1, our soil total nitrogen was highest at day 56. Now, remember when I told you earlier about how we terminated our cover crops earlier in year 2 versus year 1? It kind of explains too a little bit about what's happening here in our soil total nitrogen. So we hypothesize, or we think that our soil total nitrogen took longer to increase in year 1 because those stalks of the cover crop became really lignified. So it took longer time for those nutrients to be released from those cover crops after termination. So we see in year 1 where we had lignified stalks, it took longer for that nitrogen to be made available. However, in year 2, where we had young tissue, when we terminated, we see that that nitrogen was made available quickly by day 14, 2 weeks after termination. So our soil, our summer soil total carbon, we definitely also had fluctuations in soil total carbon. And we also found that the overall range of soil total carbon increased between 2018 and 2019. For our summer soil organic matter, our highest soil organic matter was found in the sorghum Sudan grass treatment, and it was the lowest, as expected, was in the bare control treatment. As seen with other soil properties in summer, the peak of soil organic matter was found 14 days post cover crop termination. All right, so when it comes to looking at the arthropod abundance, one of the other parameters we wanted to look at was soil moisture. There were many studies citing that soil moisture has a large, it can really predict the amount of arthropods that you're gonna see within your soils. However, we found that soil moisture did not have an effect on arthropod abundance. And also, we found that soil moisture content did not vary based on the treatment. We, thought that soils that would have more cover crops would have that protective layer of the cover crops, and even with the cover crops tilled in, it would probably help preserve some of the moisture. So the cover crops treatments would be more moisture rich than the bare control plot. But all of those hypotheses were just found null, because that's not what we actually saw when we took the samples. We found that the moisture did not vary based on treatment. However, there was a significance in day post termination when it comes to soil moisture. So we found that at day 0, the day that we terminated the cover crops, that had significantly lower soil moisture content than the other sampling dates. So our summer experiment, we had similar results. There was a significant effect of soil moisture based off of the day, just like in the winter study. Soil moisture at termination and 14 days after termination was significantly lower than day 56 after termination. However, soil moisture content did not vary based on the treatments. So you may be asking, what arthropods did we actually see? So this is a table of the family and one class and 9 families that we found within the soils. As you can see here, arachnids predominated, and so we see that we had a lot of mites, and then everything else was really lesser, which is very interesting because you would expect that you would see more arthropods living within the soils once we have these cover crops. However, that was not the case. We found that the arachnids were the ones that really predominated. So because of this, we restricted our statistical analysis to only looking at arachnids, because we didn't have enough individuals from the other families for us to really be able to include them into the statistical analysis. Oh, one thing too, I also wanted to share, we did find nematodes. However, they still weren't anywhere comparable to the amount of arachnids that we found. So as you see here in our winter experiment, our bear control plot actually had more arachnids than any other plot, which was interesting. Our results indicated that wheat and triticale vetch all had significant effect on arachnid abundance. However, the data indicates that arachnid abundance for these 4 treatments would be less than the control treatment. They were significantly less than the control treatment. And some of the trends that we found was that for every 1% increase in soil carbon, that arachnid abundance would be approximately 255 times greater. And then for every 1% increase in soil organic matter, arachnid abundance was 74 times fewer. Now, when it comes to our summer experiment, our results estimate that arachnid abundance in the buckwheat and cowpea treatment would be approximately 5 times greater when compared to the control treatment. So our buckwheat and cowpea was the treatment in which our arachnids really liked to exist. And we found that soil organic matter was the only significant continuous predictor variable for the summer experiment. For every 1% increase in soil organic matter, arachnid abundance was approximately 4 times greater. So in discussion, one of the things that we found in the literature is that arachnids do play a big role in shredding organic matter and to make it available for further mineralization by other microorganisms. So, our hypothesis was that more cover crop residues would equal more below ground arthropods. However, that was not the case. We did find that we found a lot of arachnids within the soils, and the arachnid abundance has been seen arachnid abundance has been found within agricultural ecosystems. That's well studied, but we didn't, as stated earlier, our idea that more cover crop residues would equal more below ground arthropods was not found to be true. So what we did find, though, is that arachnids actually display possibly a preference to certain cover crop types. So in the summer experiment, we found that the arachnids favored the buckwheat and cowpea treatment. Their abundance was 5 times greater than the bare control treatment. And it's very interesting because the amount of biomass that we had for the buckwheat and cowpea treatment was comparable to a couple of other treatments, such as the millet and cowpea, millet and buckwheat treatment. So what we're finding is there might be a feeding or habitat preference that arachnids have, and they might have a seasonal cycle that we don't yet understand, in which they like to predominate more in summer soils than they do in these winter soils. Because when we were testing for arthropod abundance during the winter cover crop treatment, which was after termination, so it was early spring, we found that we just didn't have a large abundance of arachnids as compared to the summer treatment. So there could be some things about that that we're still learning and should investigate. So soil organic matter, it was interesting that we had like opposite effects when it came to summer versus winter. So as we found, it was 1% increase in soil organic matter. Arachnid abundance was 14 times greater. That was similar, that study, what we found was similar to other studies in that for the summer treatment, more organic matter led to more arachnids. But then in our winter treatment, we found that more organic matter led to less arachnids. So I know in other research studies, such as the Buchanan and Hooks, their study was looking at arthropods in cucumber plots, and they saw that the arthropod numbers were reducing. And for them, they were hypothesizing that maybe the cash crop had an interaction with the arthropod abundance. And we were growing cash crops after termination of each of these cover crops, but we did not measure what that interaction could have been. So we don't know if that had an effect on our arachnid abundance. So when it comes to soil moisture, when we first started collecting data for the looking at soil moisture and arthropod abundance, it looked like there was a significant effect of soil moisture on arthropod abundance. However, once we really got to analyze all the data, it wasn't so, at least in our high tunnel system. Other research studies have shown time and time again that soil moisture has a big effect on arthropod abundance. But what we're thinking is, in a high tunnel system, we've created this little ecosystem and this microclimate that might have some effects on what types of arthropods are found, and it might have an effect on the evolutionary changes of the arthropods and the behaviors and traits that they display. So while in our study we found that soil moisture did not affect arthropod abundance, that simply could be due to evolutionary changes that have taken place, and maybe the exclusion of certain arthropod families and classes simply due to the environment that persists within the high tunnel. So in future work, we think it may have been valuable to measure residue decomposition and quantify other factors such as microbial biomass and diversity throughout cover crop decomposition. And we also think that looking at cash crop quality and yield may also give you a little bit more insight onto arthropod abundance and the families identified. In conclusion, arachnids or mites were identified as the most abundant arthropods, and their abundance was influenced by soil total carbon and organic matter. We found there to be variations in abundance of mites in different cover crop types, which suggests that soil mite abundance may be influenced by habitat or dietary preferences. And despite previous studies in open field systems stressing the interaction between soil moisture and arthropod abundance, this was not the case in our high tunnel system where we have a modified climate. In closing, we really do find that soil management practices in high tunnels have complex ramifications, possibly altering soil biology and nutrient reserves. The hope is that the data will expand the body of knowledge of high tunnel production and cover cropping by discovering their interplay in affecting below ground arthropod species and abundance. So I want to say a special thank you to the research team back at the OREC. They really did the Olathe Horticulture Research and Extension Center. I'm not sure if they changed the name as of yet, but thanks to all of those people that helped with making this project completed. And then also, of course, thank you to North Central SARE for funding this, and the Organic Agriculture Research and Extension Initiative, and of course, my major advisor at the time, Dr. Carrie Rivard. So if you have any questions, I'll be happy to answer your questions. So you can go ahead and unmute yourself if you have any questions.
Does anyone have any questions? You can drop them in the chat, or you can, I think you are able to unmute.
Yeah, actually, Scott Bard. I have a question about how you terminated these cover-ups, and did you leave them being down, or what? Did you break them up and move them to the?
You are breaking up, but I think you were asking about how did we terminate the cover crops, is that correct?
Yes, that is correct.
Okay, so our cover crops, as you can see here in this picture, these are different plots with different cover crop treatments. So when it came to termination, we had to be very careful. What we actually used were hand shears to cut down the cover crops and to then keep them in their prospective treatment plots. And then after we were done, what we did is we waited a week. What we did, well, after we cut them down, we overhead irrigation, as you can see here, these white poles. We used overhead irrigation, and then we tilled within each plot the cover crop. So that was a week later, we tilled the cover crop within each plot, and we tested from the day of termination to 8 weeks after termination. Any other questions?
That's really fascinating, Ashley. Thank you so much for taking the time to share with us. And if you're sticking around for the next presentation, if anyone has any questions, maybe they can populate them in the chat and we can reach out to you with those.
Awesome. Thank you.
All right. And next up, we have Tom Buller. Tom is the Horticulture Extension Agent for K-State Research and Extension, Douglas County, and a farmer who has been growing vegetables organically in the Kansas River Valley since 2006. He co-authored Growing Under Cover, a Kansas Grower's Guide, and Growing Over Cover, a Kansas Specialty Crop Grower's Guide to Cover Crops, with partners at the Kansas Rural Center and K-State Research and Extension. Welcome, Tom.
Great, thank you. I'll get my screen up here and get going. So I'm going to talk about a research project or research and demonstration project, I guess, that was funded through the SARE partnership grant. I'll talk a little bit more about that at the end because it is one of the grant programs that's currently open. So for people who are interested in doing some on-farm research, that's a great opportunity. So I'll provide a little bit of information on that. Unlike Ashley's research, we're on the very light end of research. So we call it evaluating and demonstrating here. So we did a project. The partnership grant is working with at least 3 farms. So did a project with 3 local organic farms here in Northeast Kansas to work on some weed control options. So the title is here, Evaluating and Demonstrating Weed Control Options for Direct Seeded Fall Crops. And the genesis of this project was really in the fall of 2017. I was talking with a number of growers who I know in this area and sort of asking them what problems were they having. And it was in the fall. So fall weeds were an issue that was top of mind for them. And specifically the problem that they had was with some direct seeded crops. So we'll get into that. So here are the partners that worked on the partnership grant. So as the extension agent, I sort of oversaw the grant, managed some of the logistics and things like that. But the farmers really made the work happen on the ground. So we have Jill Elmers from Moon on the Meadow Farm, Kevin Prather and Jesse Asmussen from Mellow Fields, and Jen Humphrey and Jess Pearson, excuse me, from the Red Tractor Farm. And so they were managing the day-to-day trials of the different weed control techniques I'm going to talk about on the farm. So here's the problem in a nutshell. In the fall, you know, here in Kansas, we get kind of a second cool season of crops. So you can start replanting the things that went in the ground in the spring. So your lettuces, things like beets and carrots, all of those cool season crops we get another shot at. But the conditions are somewhat different in that one key factor, and this is what they all noted in the fall of 2017, is that the warm soil temperatures really allow aggressive or germination of aggressive weeds. And the one that they really called out that year was pigweed. So amaranth, the variety of different subspecies in the amaranth genus, red-rooted pigweed here. Amaranthus reflux was the main one I think they were dealing with. But this is a weed that germinates very quickly. If you've had problems controlling it in the past, you probably have a pretty substantial weed seed bank built up. And so it can be a challenge to manage. And the real challenge here, you know, so in the spring, farmers don't have to deal with that quite as much because the cool soil temperatures, it doesn't really start germinating till things get warmer. So all of these crops, the lettuces, spinach, things like that, have a chance to get established before some of the more aggressive weeds like pigweed start germinating. And in the fall, they just don't have that chance. So these weeds are germinating and they can outcompete the direct seeded plants. So this is especially a problem for the systems I'm talking about. So all 3 of those farms are certified organic. And the crops we're looking at specifically have a high planting density. So if you space the rows out, you can do weed control between the rows with cultivation. But if the the plants are planted close together, it's hard to get that in-row cultivation done. And it's very easy to lose the crop to too many weeds. So here's an example. This actually isn't from any of those farms, but here's an example, of a fall cool season crop field. You can see there those tight rows of those are little lettuce and spinach seedlings, and then there's some head lettuce on the right there. But in those middle rows, especially, you can see the spacing between those crops is especially tight. It's very hard to do any kind of mechanical cultivation around that. So if those weeds start germinating and outcompete your crop, really the only options are to spend a really probably ridiculous amount of labor hand weeding the crops, literally getting down sometimes on hands and knees, or abandoning that planting entirely. So not great options. And here's the problem with pigweed specifically. This isn't a weed one. This is actually an ornamental amaranth that I took pictures of in a garden, because no one tries to let their weeds get this out of control. But weeds in the amaranth genus, they can put on up to 900,000 viable seeds. Some of those seeds can persist in the soil, able to germinate for up to 20 years. So if you have a few years or before you got the land, someone didn't control these weeds, you can really have a ridiculous problem on your hands. To wit, this is a picture from a local farm here. This is not direct seeded lettuce. Every one of those little red plants, and then the fewer larger green ones, those are all pigweed seedlings. So this was just an area that had been prepared for fall planting, and then left fallow for a few days. It got some rain, and all of these little seedlings popped. So you can get a sense here of what kind of problem farmers could be looking at if in that same interim time between the rain, they had tried to plant some seedlings here. It would be almost impossible to rescue them from this, because a lot of those seeds probably wouldn't germinate even for another few days after this picture was taken. So the solution, the farmers came to me with this sort of problem. What are the solutions to that, given an organic framework? Well, I did a bunch of reading, and the interesting thing is the fact that they're organic in this case didn't seem to matter a whole lot, because there aren't really good herbicides to control this situation on conventional farms, because you're trying to control a broadleaf weed in broadleaf crops, some of which, especially like spinach, is pretty closely related. So there aren't sort of effective selective herbicides or anything like that. But even if there were, these farms couldn't rely on those because they're organic. So what is the option? And it turned out the best option that I was able to come up with was this idea of stale seed bedding. So this is something where you prepare your seed bed like you're going to plant it. And then, just like that previous picture, you let it sit there till all of those weed seedlings germinate. And then you kill those in some way, shape, or form, ideally without disturbing the soil to the point that you bring up new seedlings into that germination zone. So weed seeds will only germinate at very specific depths, depending on the type of seed. And so if you can eliminate the weeds that are in that germination zone using stale seed bedding or another technique, you can really reduce the weed pressure over time. And this is something, ideally, if practiced a number of years, good weed management on a farm. I did say some of those weed seeds can last for up to 20 years, but the number of viable seeds go down each year. So if you can prevent the weeds from going to seed, ideally this gets easier over time. But at the beginning, if you just get a new piece of land, or if you let it go for a little bit, it can be quite a task. But stale seed bedding is sort of the top tool for dealing with that. And so the question is really, to me then, is what's the best way to do that on a farm, an organic farm? So you build the seed bed, you let all those weed seeds germinate. A lot of small farms sort of rely on tilling to kill weeds in that situation. And unless you're doing a very, very shallow depth, that tilling is going to bring up more seeds into that germination zone. And so while you're killing some of those seeds, which is always a great thing, you might not be solving the problem. So what other kinds of things could we do to maybe more effectively do the stale seed bedding? And so that was the sort of purpose of our project. What were some different techniques we could try, and ideally do that in a very way that minimizes disturbance to the soil. So not trying to promote the breakdown of organic matter, not trying to disturb those, fungal networks we heard about from the keynote more than we have to. So what are the minimal disturbance techniques? So yeah, these are the objectives. We tried this on 3 different farms. We came up with a variety of techniques, and the farms kind of got to choose the ones that worked for their cropping system. So they didn't all do the same things. And the crops we looked at specifically were beets, carrots, and spinach. I think of all of these, carrots are probably, the slowest to germinate, so maybe the most problematic. But definitely, pigweed and some of these other fall weeds can outcompete some of these other vegetable crops. So one of the goals in this, in addition to trying to figure out what's the most effective, what controls the weeds the best, we did have farmers kind of track the amount of labor time needed to adequately control the pigweed, to try and see if the different techniques we're trying here had any difference in sort of the net labor results. So not only which worked the best, but maybe which is the most economically efficient. So the trial techniques we chose, so all of these start with preparing seed beds and then trying to figure out how to kill those weeds. And so the techniques we experimented with, that is one called occultation. And then we used a flame weeder to kill off those seeds. And I'll provide some examples of these. So if you can't visualize what occultation is, hopefully I'll help you in just a minute here. And then also a power harrow. We did try one other thing that isn't really a stale seed bedding technique, but it has sort of the same goal. This is trying to give those direct seeded crops a jump on the weed competition. And so in 2019, we tried the paper pot transplanter. And so this is the idea of basically a mechanical way to plant vegetable crops very close together in an efficient manner that have germinated off-site, basically. So giving them that head start that they can't get in the field against the weeds, and then hoping that allows them to outcompete. I say this started in 2019, so we did the research trials over 2 years. The first season in the field was 2018. And when we wrote the grant, we had envisioned using the paper pot transplanter. And in between the time we wrote the grant and the time we were getting ready to start using it, the National Organic Program decided that the glue used in the paper pots was not NOP compliant. And so they did not allow, in the 2018 season, paper pot transplanters could not be used for certified organic production. Then in the fall or winter of 2018, they changed their mind, or maybe the glue changed, I'm not exactly sure. And they decided that then the paper pot transplanting system is allowable in organic production. So we got that and tried it in 2019. So we only have 1 year of sort of results from that. And just because the farmers couldn't use it without jeopardizing their organic certification. So occultation, basically this is just using something opaque to block the light out from an area. So silage tarps are the most common way I see this done. Here's a small example from a garden. This isn't one of our trial farms, but they were using bigger sheets of plastic that I found a little less photogenic, I guess. So silage tarps are often black on one side and white on the other side. You put them down, inhibiting the light. The theory is, especially if you put that down just after it's rained or you irrigate, the weed seeds that are in that germination layer are going to want to start growing. They'll pop through the soil surface, they'll be deprived of light, and they'll die. So you leave this down kind of depending on the season, between probably 4 and 6 weeks. To allow that process to occur and those weeds to die off. And then you remove that, and the biological activity underneath has ideally left you a pretty good planting bed to plant into. And so you can plant into it without necessarily stirring up additional weed seeds, sort of the ideal case. Flame weeding, we bought this Pyroweeder as an experiment. This picture, you can't quite see it, but this is one of our farmers, Jill Elmer's. We're actually at Kevin Prather's farm here, Mellow Fields, and they're just playing around with it. But walking down a row, so this is designed to straddle the size beds they're using. And there's 5 burners in the front here. And we've got another picture that I probably didn't set it to share the sound, which is hopefully good because it's just kind of roaring. But here you're walking, you can see the burner. You can see a little bit of flame coming up and some things catching on fire. But the goal of that really isn't to burn up the weeds that are on the soil surface. You want to use this when weeds are just popping. And when you use that, you don't need to ignite the weeds. So those few that went up was overkill. But what happens is the water in the plants or the water in the plant cells starts to boil basically, or expand, and that will rupture the cell walls and it'll kill plants. It's somewhat variably effective, so it works pretty well on broadleaf weeds. It doesn't work so great on grasses, but a lot of the problems they were having were with broadleaf, so it seemed like an appropriate solution. Here's the next one. This one's called a power harrow. And so it's this implement behind the tractor here. I'll play this video too. So you see something moving there, and basically what's happening with this power harrow, unlike a rototiller that has blades or tines that move sort of along a horizontal axis, this has a vertical axis. So they're just spinning around in circles, not bringing up new weed seeds. So they're not mixing the soil layers. So you get a nice, prepped seed bed there, like you see, that looks very similar to if they had run a rototiller over that. But they didn't introduce those new weed seeds. So the idea is, in this stale seed bedding technique, you prepare the seed bed, you let those weeds germinate, and while they're little, you get over them with the power harrow, kill those weeds, but you don't introduce more weed seeds. And then, yeah, here's another view of it. So this is just from the tractor. You could see, I don't know why it jumped ahead there. You could see it's just, it doesn't like that. Anyway, it's just mixing the soil in sort of a horizontal plane rather than mixing it vertically. And so this is the last thing we tried. I should have gone back and taken a picture of them using it, the paper pot transplanter, but this is just from the website. So this, you plant the seeds in little paper pots that fit in more or less a standard 10-20 tray. And so in one of those trays, there might be 200 seedlings. So they end up being spaced. You can buy the spacings on this paper. It's a paper chain that's tied together, folded up into that tray, and then it unfolds as you drag this across your garden bed. And so you can buy those in different spacings. So you can get 2 inches, 4 inches, or 6 inches. So this really isn't, again, stale seed bedding, but this is allowing, that dense planting we're looking for with some of these fall crops, but getting an early start on it so that they can outcompete the weeds. So the results of trialing these different techniques in 2018, Farm 1, they tried all of these techniques, and then we had what we call, just a control where they basically did whatever they normally would have done, whatever kind of weed control they normally would have done. They did that, most of which was, you till it up, you plant it, you go through and you hand weed it. In Farm 1, in year 2018, there was no significant difference in labor time. There was no difference in weed control for either of the 2 treatments they tried, the power harrow or the flame weeding. And we'll get to why they only tried 2 in just a minute here. Farm 2 only did occultation, and they had, even with that technique, just totally inadequate weed control, both in the zone they did occultation in, and then the control zone where they hadn't done that and they had just tilled. The weeds just got away from them. So it was to the point that they abandoned both of the crops. So it didn't make a significant difference, I guess. And then the farm 3 did have a significant difference in the performance of the two techniques. Occultation seemed to work the best. The power harrow as the weed control and the stale seed bedding worked second best, and the control the last. And so they did not do the flame weeding. They just did the power harrow and occultation. And so why didn't farm 1 do occultation? So here was one of the challenges, and this was really a challenge even with the things they did. This is just sort of their timeline in the fall. So in July, they prepped their beds and covered them with the tarp. And then on August, they got the rest of their beds ready. And then mid-August, a month after they'd put that tarp down, they uncovered them. And the 18th and 19th, they fertilized and planted all the beds. They did the power harrow over the beds that needed power harrowing. And then they planted those. And then for all of those trials, they got 0 germination on any of the weed controls or even their control. They just didn't have good enough germination. So they abandoned all of those crops. They did replant. So for the power harrow and the flame weeder, they replanted and were able to get some results for that. But since the occultation takes a month to do it, you know, kind of if you try it and it doesn't work, you can't try it again because, you know, a month after September, it would have been too late to plant most of those things. So we didn't get occultation results. I mean, here's what was happening in 2018, in case any of you didn't remember. In Northeast Kansas, we were having a pretty epic drought. So the soils were hot and dry. So even though they tried to irrigate to bring down the temperatures a little bit, those cool season crops just did not want to germinate. So they didn't establish a good enough stand. And that's one of the challenges with these kind of demonstration projects. I mean, they sound good. And then, you know, especially if you try and run them in a consistent way or using more sort of rigor in who gets to do what, you know, every year you get a curveball thrown at you by the weather. So yeah, here's some visual pictures. It's not that easy to tell here, but this is all spinach. So the taller green things you see are spinach seedlings. The littler green things you see are weed seedlings. And because this whole trial, so this is that farm that had to do the replanting, because that whole delay basically set them back more or less a month on their fall cool season crop production. They lost the weeds that we were trying to control were kind of out of the system by then. So all these little weed seedlings you see is actually henbit, which is a winter annual weed, not sort of an aggressive summer weed. But anyway, you can get a sense here for there was some variable weed control. But unfortunately here, when I line these up, this one on the left here was the one that was done with the power harrow, the flame weeder, and then the control, which in theory should have had the worst weed control, actually looks the best. They did go through and clean up all 3 of these beds just by hand with a hand hoe, because even the control doesn't have adequate weed control. And basically, because they had to cover that whole field, they reported that there wasn't any difference in the amount of time it took them to do each of these 3 different planting beds. So yeah, there's a larger picture. So these were 3-foot by 100-foot rows, more or less. And here's a little close up of what it looked like in row. You can see there, this is a little bit later, so the spinach is a little bit bigger, but there, those little pinkish leaves and some of the little rounder green leaves, that's all henbit. So we aren't really struggling with any of these real aggressive weeds. Henbit will stay around throughout the winter, but it really actually doesn't outcompete spinach very effectively. So it's hard to say, you know, whether this worked, because that delay meant that the weeds we were really targeting were already done, but we didn't have any very impressive results on that. Here's farm 2. This was for a field day we had, so you see some signs there marking out, and there are carrots in there, I promise, but they're hard to find. So they really didn't get the weeds controlled effectively. So where you see the tarp on the right there, it had been over this area prior to planting these carrots. Then it was moved. That was their next bed that they were going to use it for over the fall and winter. So really not adequate control in either system. So 1 year of occultation didn't really do it in this farm. There are carrots in there, yeah. And so this is just the sign we had up at our little field day. And then here's the third farm, and I got these pictures a little bit later, so it's a little harder to tell. But this one did have some pigweed competition issues. They didn't have the issue getting their crops established in the fall. I think they started a little bit later, so the soil had cooled down a little bit. But they did still have some pigweed. Unfortunately, I took these pictures after frost, so all the pigweed's dead. But what you can tell is more or less by looking at the amount of debris in the rows. So you can see here, I'll put up, this is the control on the left, and you can see in the row there and in the alley, there's a lot of what looks like straw mulch. Well, that's actually all dead weeds. So there were a lot of weeds in there that died back when it froze. These two rows were done by the Power Harrow, and they are significantly cleaner. And then this third row was set aside a little bit, and that's where the occultation was. And you can just see there, this is significantly cleaner than the row on the left or the row on the right, which is a different crop, but planted at the same time, and it wasn't in that occultation zone. So that technique seemed to work well. The Power Harrow seemed to work well. At least both of those worked better than the control. There wasn't really a significant difference between the two of them. In 2019, so the first farm, the farm 1 and farm 3, I guess, added in the paper pot transplanter. They took some experimenting, so they started using it in the spring, not as part of this trial, but just more or less to get used to how to use it as a system. So they had some learning to do with that, specifically how long can you leave things in those paper pots. You can't leave them in there as long as you normally would with cell transplanting, because the paper pots are kind of open on the bottom and they're just a chain. So what happens if you leave the plants in there too long, they'll grow out the bottom and their roots will merge, and then that chain doesn't unwind as you pull that through the soil. So you need to be ready to move those pretty quickly. So for carrots, a couple weeks after they germinate, if you leave them too long, they'll get a big taproot and get tied up. The challenge we had in 2019, they planted the fields, things looked good, and then the deer ate all their produce. And they're next door to another farm, one of our other trial farms, also lost a lot of their produce to the deer. So they did use a silage tarp. It was on for 4 weeks. It did a good job killing the weeds. So the occultation worked. But the challenge they had this year is we did have some heavy rains over that time. And so that tarp, they had prepared the seed beds, so nice, light, fluffy soil, put the tarp down, you get heavy rain, water standing on top of the tarp, and these nice, light seed beds they had got compacted by the weight of that water. So they weren't really able to plant in those directly. So they actually drove the Power Harrow over it, which kind of mixed up the experiment results, but they needed to get their crop in the field. And that was a decision we made at the time that, yeah, go for it. And that worked really well in terms of taking that compacted area and loosening it up again. They had some nice spinach that year from that bed, and not good spinach from the bed that they didn't do weed control on. Farm 2 couldn't participate. And then farm 3 that year, due to the heavy rainfall that caused that compaction at the other place, they didn't get their fall crops successfully established. So in any of those, the control or any of the stale seed bedding techniques. So it was really hard. I mean, as you can see here, we have a lot of information, but not a lot of data to back up what happened. But we do have some observations. One thing they've found, the single methods don't seem to work effectively to control weeds, but doing a couple of methods seemed like that worked pretty well, like following occultation with the Power Harrow. The paper pot transplanter did kind of work as advertised to get those plants established above or ahead of the weeds, so they didn't have any weed control issues. But both of the farms that experimented with it actually loved it, and way beyond issues of weed control, just like the way it helped them manage their production. So we bought one as part of this project for them to share, and they decided they went out and bought another one so they don't have to share it because they both use it all the time now for all these kinds of crops. So yeah, I thought that that was maybe the takeaway, because this research project was in 2018 and 2019, so we're a couple years on. So, you know, some of the results weren't that great, but based on their experience, what are the farmers still using? And I take that as a real sort of indication of what's valuable. So they're all, or actually two of them, one of them has actually gone out of business. But the two of them that are still in farming are both heavily reliant on occultation. They use a lot of tarps. There is some learning curve with that besides the compaction, in terms of whether the white side or the black side is the best at what time of year, how long to leave it out there. There was some indication maybe that if you use the black side in the summer, it actually got so hot that it kind of shocked the seeds in the ground into dormancy rather than them germinating and dying. And then when you took it off, they would all germinate. Still experimenting with that, I think, from what I've heard from them. They both like the power harrow and use it a lot to create seed beds without bringing up additional weed seeds. And they both use the paper pot transplanter for lots of purposes. The one thing I guess I wish I had known before we designed this study, after we got this project going, I saw a great presentation from, I believe his name is Mark Williams out of University of Kentucky, where they did experimenting on stale seed bedding. But their experiment, rather than trying 3 different tools to create that stale seed bed, they did experimenting on how many passes to go through. So they basically would do stale seed bedding for 2 weeks and then run through and kill everything. And then they did another one where that happened twice. So that one was ready 4 weeks ahead of time, got ran through at 2 weeks, and again at 4 weeks, and then one at 6 weeks. And what they determined is really the beds that they got those 3 passes on were really the most successful, and they had like 95% weed control on those. And the one that was 1 pass was about 70% weed control, which sounds really impressive and not like that much of a difference. But when you think back to that picture I had earlier about all of those pigweeds that were just like a carpet of them, if you kill 70% of those, you still have a ton of weed, like an unmanageable amount of weeds in your field. So really trying to figure out how to get to that 95%. And I think the secret was probably not to try different tools. It should have been to try and figure out how many times we need to use them to get through there, how many iterations of that before you really knock down enough weed seeds in that seed bed to consider it really officially stalely seed bedded, I guess. So concluding thoughts, just like everything in farming, weather really threw us some curveballs. So we try to be flexible to help our farmers out. Hopefully that worked for them. And the other thing with each of these tools, so we added occultation, farmers weren't doing that at the time, added this power harrow, added the paper pot transplanter, added, all these different tools. Each one of those they have a learning curve. And probably should have built in more time to figure that out. The paper pot, we got in a year late. So we did have kind of a spring to figure it out before we were using it on the trials. But that kind of not allowing enough time for that learning curve was a challenge. So anyway, with that, I think I'm open to any questions. I did want to just throw in a mention here of the SARE Partnership Grant Program. So that is open right now. Applications are due October 21. So if you and a couple of other farmers have a problem or an innovative solution, feel free to go and check that out and maybe write a grant to help you figure out how to do that.
Great, thank you, Tom. Are there any questions, or would anyone like to throw them in the chat and we can read them? Oh, Allison, did you have a question?
I had a question. I finally figured out how to how to do this. We were given the opportunity to work with a commercial plot, not a very large plot, but one that had been neglected for several years. And we didn't entirely, I mean, to some extent, we understood that there was Johnson grass there, but we didn't understand. And we would any of these things be helpful? Because in the end, we ended up having to abandon it. We used quite a bit of we used a heavy duty landscaping material to try to get it, under control. But in the end, it just pushed it up out of the way. And would any of this help with that?
Yeah, I would say of all of these, with a perennial weed like Johnson grass, the solution is really time. So if you wanted to just throw a occultation tarp over it, that would help. So the landscape fabric allows water through, so the Johnson grass wants to keep growing, whereas the darkness, it doesn't really it doesn't grow that much. And I've used it on patches of Johnson grass, but to really manage something like that, like you're not talking about leaving it down for 6 weeks, it'd probably be like a growing season. The other option I've heard in sort of organic Johnson grass control is really, a little bit of tillage is bad for Johnson grass because it'll cut up the rhizomes, and instead of 1, then you have 10 or however many. But it's doing that repeatedly. So it's really like there's not a great soil health solution that I know of, but going through there, letting it sprout as it starts growing, tilling it again. And if you do that repeatedly, that's had some success, I think, in treating that. But again, you're looking at like repeatedly for, months, if not a whole growing season. Yeah, Pantaleon's got a suggestion there. I know he's dealt with some of that. So broad forking out the rhizomes can be useful. Anytime you can get those rhizomes out of the ground, that's a win. But depending on how big of an area it is, I know it was with quackgrass, but some folks up in Minnesota had good success not tilling with a tiller, but using like a chisel plow, doing kind of the same thing like Ponta's doing with the broad fork, bringing those rhizomes up to the surface. They would die out, especially if you can do that in the fall, bring them up to the surface so they die out. And then, try and get them off before you do other tillage. So there's a number of options. Unfortunately, no really good or easy one for that, for Johnson grass.
Well, and since it's community owned, it's really hard to decide to make the investment in it because we don't know for sure if we're going to be there next year.
Yeah.
But it is going to be an ongoing problem there for whoever ends up managing that plot, or if we get moved around. Are you somebody that can come out to a community space like that and
where
I'm in Lawrence.
Oh, absolutely. Yeah.
Yeah, I'm in North Lawrence.
Okay. Yeah, absolutely.
Okay, we can visit about it.
Yeah. Let me put my email in the chat here.
Sure. I've got you.
Okay.
Is there another question, Caitlin? Because I have another question. Go for it.
Yeah, I don't see any others.
Okay. So when I was working in a different situation, I had a lot of pigweed, and we were eating it, because it is really nutritional, and also feeding it out to animals, horses and chickens. Is there a place in the market to expand that commercially? Is that something that that could be done? Why hasn't that been explored?
I think that's interesting. I think there could be, if you want to try and market it. I don't think most people would know what to do with it, but I mean, greens in the summer in Kansas are pretty hard, so it would fill kind of a unique niche in that sense. You know, I have a small farm, and I've had problems with pigweed. And on 2 separate occasions over the course of a few years, and this was over a decade ago, I looked out my window and there were groups of immigrants, one time from Southeast Asia and one time a group from Honduras, that like drove by, saw my pigweed and stopped, and they were like picking it. I was like, take it all. But you know, so it's definitely edible, very nutritious, but I haven't really seen anybody do anything with it yet. But you know, that's not to say you couldn't be the one, and I'm sure you'd have a lot of enthusiastic support from other people who want you to take their pigweed.
Okay.